Hello and welcome to this video in the series on integrated pest management, focusing on disease control in cereals. This episode is in 3 parts, each dealing with one of the mechanisms by which choice of cereal variety can help with disease control. If you're being attacked, you have 3 options. One is to try to escape, the second is to resist, and the third is to tolerate what's happening to you. And crop plants, when they're being attacked by fungal pathogens, use all 3 of those tactics to defend themselves. So let's take a look at each one of those tactics in turn in a bit more detail. In this video on integrated pest management, we're going to take a look at how choice of variety can help reduce the losses caused by diseases. And this subject's going to be dealt with in 3 parts. In this video, we're looking at how disease escape can reduce the number of spores arriving on the upper canopy of cereal crops, particularly of splash-borne diseases. In the next video, we'll take a look at disease resistance and how that can reduce the amount of disease that's caused by the spores that do arrive. And in the third video, we'll look at disease tolerance mechanisms by which the crop can reduce the impact of a given amount of disease severity on yield. So we saw in the video on how to predict epidemics how the position of sporulating lesions in the crop canopy can be an important determinant of the amount of disease that subsequently develops. We saw that in the case on the left, where at the time the flag leaf is emerging at the top of the canopy, if the nearest infectious lesions are low down in the canopy, then it's going to take a lot of intense rainfall and rain splash to move those spores up, and it's therefore much less likely that there will be a severe epidemic. Whereas if we're faced with the position on the right, where leaves are containing sporulating lesions that are right up and adjacent to the emerging leaves, then it's going to need very little rain, or indeed just some surface moisture and movement of the crop, to spread spores from those lesions onto the flag leaf and create a severe epidemic. And we also saw in the video on how to delay and reduce epidemics why it is that if we can reduce the number of spores that are arriving on the upper canopy, in other words, reduce the initial inoculum, that will have the effect of delaying the epidemic, which would obviously be beneficial. Now, unfortunately, plant breeding during the 60s and 70s had rather the reverse effect to this. If we go back on the left of this image to old varieties such as Maris Widgeon, they were very tall, and that effectively reduced the movement of spores up the canopy. We then, in the early 1970s, had what was really a breakthrough in plant breeding, Maris Huntsman, which was somewhat shorter, produced less straw and more grain, and had about a 15% higher yield as a result of that higher harvest index. And then the really big shift came in the late 1970s with the first of the semi-wheat varieties, Hobbit being the first, where a semi-dwarfing gene was incorporated into the varieties. This shortened the stem further, improved the harvest index, enabled the crop to stand up reducing lodging risk and enabling it to deal with higher nitrogen inputs. And the benefits of this to yield meant that these semi-dwarfing genes were incorporated in varieties worldwide, and most of the global wheat area is down to varieties that contain these types of dwarfing genes. And the first variety, Hobbit, was followed quickly by others, Virtue, for example, Longbow and Norman, and that led to varieties such as Riband and Consort, which were highly susceptible to splashborne diseases in general and to Septoria tritici in particular. Now, at that time, there was also the introduction of highly effective fungicides, and that allowed us to deal with the subsequent epidemics of Septoria tritici. So the ability to convert higher nitrogen inputs and higher fungicide inputs into yield responses led us into a period of high input and high output agriculture. Now, it's quite difficult to separate out the extent to which the disease susceptibility of these varieties was due to the height reduction or due to disease resistance genes or the lack of in these varieties. But fortunately, around that time, a very useful set of what are called isogenic lines were developed. So in this particular case, a variety Mercia was produced which had no semi-dwarfing gene in it, and then it was crossed with a wheat line which contained one of the semi-dwarfing genes, for example RHT2, which stands for reduced height gene 2, and you can see the effect of that in the pots here. And the progeny of that cross were then back crossed to Mercia, typically 6 or 7 times. So what you end up with is effectively the variety Mercia, but with a difference in the dwarfing gene and the vast majority of the remainder of the genome, and therefore the vast majority of the resistance genes, identical to the Mercia background. And the same process was done for a more severe dwarfing gene, RHT3, and an even more severe dwarfing gene, RHT10. So what this gave us was a set of lines where we could separate out the effects of height from the effects of disease resistance. And when we grew these lines in replicated plots in the field, this was the outcome. So if it was grown in a site where there was moderate or high disease risk, the Mercia without dwarfing genes on the left looked something like this, whereas the extreme dwarfing line RHT10 looked something like the image on the right, and the difference is entirely due to Septoria tritici. So what this allowed us to do was to quantify the relationship between height, in this case height to the base of the flag leaf, and the amount of Septoria tritici symptoms in the crop, in this case measured as a symptom area index. And what we can see is the Mercia without dwarfing genes on the bottom right, with greater height to the flag leaf and lower symptom area. The semi-dwarfing gene RHT2, somewhat shorter, somewhat higher severity. And if we went shorter still to RHT3 or RHT10, then the effect was very powerful indeed. And in this case, the effect of the semi-dwarfing gene was not that big because Mercia, even without that gene, was a relatively short variety. Now, we're also able to explore the interaction of crop height with rainfall by using rain shelters, and what we were able to show was that with shorter lines, even in virtually the absence of rainfall or just low amounts of rainfall, we're still able to create quite severe epidemics, whereas the taller lines in the absence of rainfall were pretty much healthy. Now that would seem a good argument for breeding taller, but of course there are downsides of doing that. One is a tendency for harvest index to decline, and also the risk of lodging. So as we go taller, the weight of the ear exerts stronger leverage on the stem base and the root system, and that increases the probability of either root lodging, where the root system's anchorage in the ground fails, or stem lodging, where the stem buckles. So there's a trade-off here. However, what the work allowed us to do was to quantify that trade-off. So on the left was the data we've seen, where we could look at the effect of height on disease severity in different environments. And on the right is the outcome of a set of model calculations based on field experimental data, where it was possible to quantify the effect of stem height on the probability of lodging. So what this enables is a move towards what's known as the design of an idiotype, where the design of wheat can be optimized for a given environment, accounting for these types of trade-offs. So what can we conclude from this? So disease escape reduces the arrival of spores on the upper canopy, which delays the epidemic. Taller varieties reduce the severity of splash-borne diseases, such as septoria, or in barley, net blotch or Rincosporium. But taller varieties also have higher leverage, which increases lodging probability. Nevertheless, breeding varieties shorter than we have at the moment would be very risky from a disease control perspective. In the field, the distance between the highest sporulating lesions, which are the source of inoculum, and the emerging flag leaf is a good indicator of the risk for splash-borne diseases. And we'll consider the effects of disease resistance and tolerance in subsequent videos. And there's further reading available. In this video on integrated pest management, we're going to take a look at how the disease resistance of varieties can help reduce disease severity and how we can better exploit that. So the effect of host resistance is to reduce the proportion of spores that manage to successfully infect the plant, and for those that do manage to infect successfully, the host resistance can slow down the formation of disease lesions, and the combined effect of that is to reduce the growth of the epidemic. Now that effect is pretty much the same as we obtain by applying a fungicide, which reduces infections and slows lesion formation. And we can see that in this data here from a field experiment where we applied different levels of fungicide, either 0, quarter dose, half dose, or a full label dose, and measured the severity of disease over time. And we can see that with increasing dose of fungicide, the epidemic is increasingly slowed. In that same field experiment, we also measured the effect of host resistance. So we picked 4 varieties with different levels of resistance against, in this case, Septoria tritici, and we can see that the effect of those different levels of host resistance from the 4 varieties is very similar to the effect that we obtain from a single fungicide treatment. Now, you might notice that this is data from 1995, so we're looking back nearly 30 years here, and there's a reason for doing that. The variety which is most susceptible, ribboned, is actually the variety that was most popularly grown around that time, which seems a bit odd, but in fact ribbon was a very high yielding variety with very good agronomic characteristics, and farmers were generally willing to apply quite intensive fungicide programs to control septoria in order to make use of its other attributes. Whereas some of the other varieties which had better septoria resistance sometimes had weaknesses in resistance against other diseases, or not so beneficial in terms of their agronomic characteristics or their yield potential. So we got into a phase in the 1990s of growing quite disease susceptible varieties and applying quite a lot of fungicide in order to control disease. Now, that situation has changed very markedly in the last 30 years, and it's illustrated by this data from an AHDB survey carried out in 2023 of farmers and advisors. And the question asked was, what are the features that are most important in a variety? And I think for the first time, pest and disease resistance came out as the highest priority trait for cereal varieties, coming out ahead about agronomic characteristics and indeed coming out ahead of fungicide treated yield, whereas previously fungicide treated yield tended to come out as the most important trait in a variety. And this importance of disease resistance is reflected in the amount of effort that goes into quantifying the disease resistance of varieties in the AHDB recommended list. So this is data published every year from multiple field trials across the UK where disease levels are scored, and that's translated into a resistance rating in a range from 1 to 9, where 1 would be hideously susceptible and 9 is virtually immune to that particular disease. If we turn now to survey data, so this is the Defra Pest and Disease Survey that's carried out every year. It's a survey of over 250 commercial wheat fields each year, and the data on the left show that the varieties that have been selected by farmers in those fields have been of increasing disease resistance over the last 20 or 30 years. In this case for Septoria tritici, but there have also been improvements in resistance against powdery mildew and the rusts. And there seems to be, although it's difficult to separate cause and effect, a bit of a trend that we have perhaps 2 or 3 years on the trot with quite severe septoria due to weather conditions, and more resistant septoria varieties are picked in the years following. If we delve a little deeper into the same data set, on the left here we're showing the amount of Septoria tritici in those sampled wheat fields over a 5-year period. There was a 5-year period of quite severe Septoria epidemics due to conducive weather. So this is data from well over 1,000 commercial fields that were almost always fungicide treated. And what we can see is that in those high disease seasons, growing varieties with moderate or low resistance ratings around 4 or 5 was actually quite risky, and even with fungicide treatment the level of disease was still quite substantial, whereas in the lower disease seasons such as 2015 to 2019 we could obtain really quite effective disease control even with relatively poor variety host resistance. And that's reflected if we look a bit further into the level of disease control that can be obtained with different types of fungicide program. So this is data from 2005 to 2019, so several thousand wheat fields represented here. And we've split the data down into fields which received what we're terming a high efficacy fungicide program, as opposed to what we're terming a medium efficacy program. And the high efficacy programs were characterized by more applications with typically a higher dose per application and typically more effective fungicide products being used, which also tend to be the more expensive products. And in contrast, the medium efficacy programs tended to be both less intensive and perhaps have fewer of the more expensive and more highly effective fungicides. But what we can see here is that the high efficacy fungicide program provided good control almost regardless of the level of host resistance. But if we were growing varieties with ratings of 6 or 7 against Septoria tritici, we were obtaining really effective control with fungicide programs of medium efficacy. But when we look at the data, we're still not really making sufficient use of this host resistance and having sufficient confidence in it to reduce the intensity of the fungicide programs that we're using. And to understand why that is, I think we need to look a little more into the different types of host resistance. So there are mainly two types of disease resistance, and the first of those we can characterize as being partially effective host resistance, which has a quantitative effect on the epidemic and slowing it down. This type of resistance is usually coded for by a combination of what we might call minor genes, which have been accumulated into a variety by plant breeders. And this is the main type of resistance against the necrotrophs, in particular against Septoria tritici in wheat. It usually results in AHDB resistance ratings around 5 to 7. And the key thing about this type of resistance is that it's actually quite difficult for pathogens to overcome it. So any loss of effectiveness of this resistance is usually gradual over a run of seasons. It tends not to happen suddenly. And this type of resistance can also provide valuable control against rusts and powdery mildew. In contrast, the second type of resistance, which we're calling here major gene or qualitative resistance, it's usually coded for by a single disease resistance gene of large effect. It's mainly used against the biotrophic pathogens, the rusts and the powdery mildews, and it can provide a really high level of resistance, close to immunity. So an AHDB resistance rating of 8 or 9, for example. But the downside of it is that new virulent pathogen strains can lead to quite a rapid loss of effectiveness. And if we understand the mechanisms, we can understand perhaps a bit better why this should be. So this is an example of a major gene type of resistance. It's a gene called MLO, which provides resistance against powdery mildew in spring barley. And we can see here an image taken down a microscope of the leaf cells on the leaf surface or epidermis of a spring barley leaf. So in the orange circles we can see some white blobs, and those white blobs represent where a powdery mildew spore has landed and tried to infect, and the plant cell has responded very quickly and put up a physical barrier by thickening the cell wall to prevent that infection, and the thickening of the cell wall is shown up in those little white blobs. But occasionally a spore will beat the plant to it and manage to infect a cell, and the plant then has another line of defense, which is to initiate what's called a hypersensitive response. So the plant detects that the infection has happened in a particular cell and causes that cell to die. And by the cell dying, it also causes the infection to die out, because biotrophs rely on having a living cell to be able to live themselves. Now, MLO is an unusual case because it's proved to be durable and has continued to be effective for decades. But that's not always the case with this type of resistance, and particularly when it comes to resistance against the rusts. And the reason for that is that for this type of resistance to be effective, the plant has to detect very rapidly that a pathogen is present on its leaf surface and kick in these resistance responses. And it's rather analogous to how the human immune system detects the COVID virus. And what we saw in the COVID pandemic was that the virus would change its spike protein in a way that meant the human immune system could no longer recognize that it was a pathogen. And unfortunately, fungal pathogens are very adept at doing this as well, meaning that the plant no longer recognizes that there is a pathogen knocking on the door, and it therefore fails to trigger the resistance response. And the effect of that can be seen in what happens to the resistance ratings of varieties after they're introduced onto the recommended list. And this is a set of examples for yellow rust of wheat. This is for a whole range of varieties introduced in the 2000s. We can see that varieties are introduced onto the list with a particular resistance rating, and sometimes that resistance is maintained at a very high level throughout the life of that variety. So some of the lines along the top are maintaining close to immunity for many years. But in other cases, the resistance is subject to a very sudden fall, sometimes within one season, when a new race comes along that that particular type of resistance gene can't recognize. So this introduces a kind of uncertainty into the amount of trust that can be placed into this type of disease resistance, and therefore introduces a sort of uncertainty into whether we need to treat yellow rust with fungicide. But on the positive note, new virulent strains are tracked and published by the UK Serial Pathogen Virulence Survey, the UK CPVS, and also in recent years varieties that have suffered a significant loss of resistance tend to be removed from the recommended list more promptly than perhaps they were going back into the 1990s, for example. So the other aspect of this type of major gene resistance is that it doesn't always kick in all the way through the life of the crop. So this is data taken from the 2024-25 recommended list for resistance against yellow rust. I've pulled out here the varieties which have a resistance rating of 9, so close to immunity against yellow rust. But what we can see is that some of these varieties are resistant throughout their life at the seedling and at the adult plant stage, whereas a couple of these varieties are actually susceptible at the seedling stage but become resistant as they become more mature and as they move towards emergence. So we need to be aware of this in the varieties when we're deciding on our fungicide programs and the degree to which we need to treat specifically for yellow rust, or where we can rely on fungicides applied to control other diseases to also provide sufficient control of rust. So what can we conclude? So the nice thing now is that we have really good disease-resistant varieties available in all the wheat quality groups, and those disease-resistant varieties are providing very high yield potential and also good agronomic traits. So this is a major success for plant breeding. But I'm not sure in the field that we've adjusted sufficiently our fungicide inputs to account for that improved disease resistance of varieties. And part of that, I think, is driven by uncertainty about new virulent strains, particularly against the rusts. But if we pay attention to the serial pathogen virulence survey, I think it's less likely that we'll be caught out by those sudden changes. And the resistance against septoria is generally highly reliable and not subject to sudden changes. And one of the main reasons for this success in plant breeding has been due to advances in DNA sequencing and very high throughput markers that can be used in the breeding program to identify whether particular breeding lines carry disease resistance genes. And that has helped plant breeders to stack minor resistance genes into varieties, thus improving their effectiveness. And it's also enabled minor genes to be included in varieties that also carry major gene resistance. And that means it's less likely when the major gene breaks down that the drop in resistance will be as severe as we used to see in the past. And the other bright point, I think, for the future is the Precision Breeding Act of 2023, which provides a route which can permit the use of gene editing, which can be useful for introducing host resistance genes, although there are quite a few hurdles still to get over before I think we see the benefits of this, not least in the question of whether this type of technology proves to have public acceptability. And in the next video in this series, we'll take a look at a different aspect of the way varieties cope with disease that doesn't rely on disease resistance genes. And if you want to read more on this subject, there's a whole range of references here. In this video on integrated pest management, we're going to take a look at how the tolerance of varieties to disease can reduce the losses caused by disease. And we need to be clear right from the start that Although tolerance is sometimes used as a synonym for resistance, here we're using tolerance in its strict sense to mean the ability of a variety to maintain yield despite the presence of disease. So the first question is, if we have good escape and we have good resistance and also we have effective fungicides to reduce disease, why do we need tolerance? And there are 4 reasons for this. The first, as we saw in an earlier video, is that the type of host resistance of varieties that's most durable and likely to be reliable is a form of partial disease resistance, so it's not providing complete control of the epidemic. Secondly, as we saw in another video, the margin economically over fungicide cost is maximized with some disease left in the crop. It's simply not economic to remove all disease from the crop canopy. The third point is that even if we could achieve that, it wouldn't be a very good idea, because the harder we try to control disease, either through host resistance or by the use of fungicides, the greater the selective pressure we put on the pathogen populations to evolve virulence and resistance. Hence the phrase, don't be too keen to be clean. And the last point is that tolerance has certain advantages over resistance in that there's really no mechanism by which the pathogens can overcome it. So it should be very durable, and also tolerance mechanisms should be quite broad in their applicability across all the major foliar diseases. So let's take a look in a bit more detail at what tolerance is. We saw earlier in the video about how long the canopy needs protecting for. This diagram of how the green area index of the crop canopy increases through time, reaches a plateau, usually around ear emergence, and then declines, and how that decline is accelerated in a crop canopy that's diseased. And we discussed that if we add up all the area of green area index under this curve, we end up with a measure of canopy duration, which we're calling healthy area duration, or HAD. Now, if we go to a set of replicated field trial plots with a range of varieties, and we measure their healthy area duration over time like this, and we do that in plots which have been very intensively treated with fungicides, so they're protected from disease and healthy, and in other plots that are either less well protected or not protected at all, so they become heavily diseased, then what we can plot is a chart like this hypothetical example here. So on the bottom axis we have the healthy area duration, and up the vertical axis we have the yield that we measure. And clearly the more disease there are in the plots, the smaller the healthy area duration. Now when we do this, what we find is that varieties differ significantly in the slope of these relationships. So those varieties or breeding lines that have a shallow slope are exhibiting high tolerance because they're losing less yield per unit of HAD loss to disease, whereas an intolerant variety is going to have a steeper slope and lose more yield for a given amount of disease. And about 20 years ago, we did this very intensive set of measurements in order to quantify tolerance for a panel of about 25 of the then currently elite winter wheat cultivars in the UK. And when we plot them on this chart, this is the scatter diagram that we get. So in this case, along the bottom axis, we've got the yield that those varieties achieved where they were very intensively protected against disease. So we can consider that as a measure of their yield potential in that particular environment. And up the vertical axis, we have the tolerance of the variety. And you'll see that the numbers get bigger as we go down, because as we saw, the steeper the slope, the lower the tolerance. So the varieties on the top of this diagram are exhibiting good tolerance, and the varieties at the bottom are exhibiting poor tolerance of disease. So the first thing we showed was that there was significant variation, meaning both significant in terms of its value to growers, but also significant statistically. But the other trend that we saw, albeit a little weak, was that it tended to be that the varieties with the high yield potential tended to have somewhat lower tolerance. And we'll come back to that point later on. So let's have a think for a moment about how this difference in tolerance might come about. In the video about which parts of the canopy need protecting, we looked at this slide where we defined the source of carbohydrate production in the plant as being a combination of the amount of solar radiation received and the proportion of that solar radiation that was intercepted by green crop canopy. So that determines the amount of carbohydrate and to some extent protein that's produced. And on the other side of the equation, we've got the sink capacity to store that carbohydrate and protein, which is determined by potential grain weight, the number of ears per meter squared, and the number of grains per ear. And the more of those we've got, the greater the capacity for storage. Now, what we then did was to take hundreds of breeding lines and measure their tolerance and measure their source and sink characteristics to see if we could identify particular characteristics of those breeding lines and varieties which were associated with good or poor tolerance. And over a set of studies and including some modelling, what came out was this list of traits which we found to be associated with disease tolerance. Now, we don't need to go through each of these in detail, but in broad terms, most of these traits were showing increased source capacity or decreased sink capacity. And perhaps it's not surprising that those things would be associated with tolerance, given that the effect in wheat of disease is to reduce source capacity. Hence, if we've got, as it were, a bit of spare source capacity, then the crop's in a better position to withstand the effect of the epidemic. And a good sort of overall measure of what was associated with tolerance was this combined trait of healthy area duration per grain, which again is demonstrating this impact of source-sink balance on tolerance to disease. Now, if we then take a look at the data that we collected, so this is data where we have taken two varieties or two breeding lines and we have crossed them and we have created hundreds of offspring, and those offspring have been through a process of double haplodization been made through breeding. And for each of those lines we measured their potential yield under full fungicide protection, and we also measured their tolerance. And this is the pattern of results that we got. and statistically we can describe this pattern of results by the solid line that surrounds the points, and effectively that solid line represents the range of potential yield and tolerance that was achievable within those hundreds of progeny of wheat crosses. So this is the kind of plausible breeding population from which we can operate. Now, what you can see is that, again, the very most tolerant varieties tended to have somewhat lower potential yield, and the varieties with the very highest potential yield tended to have somewhat lower tolerance. So there's clearly a trade-off here, that if plant breeders breed for tolerance within what's plausible, they may be sacrificing some potential yield in order to do that. And the question is that really worthwhile? And this is just one slide for those of you who enjoy an equation, and it's by way of explaining how we address that question. So we start at the top with the first diagram that we looked at, which was the hypothetical straight line diagram of health area, duration and its impact on yield, and the slope representing the tolerance of two hypothetical varieties. And the equation to the right of that diagram describes that straight line. So for any level of had that we manage to achieve in reality in a crop, and for any given potential yield, we can calculate from this straight line what the actual realized yield would be. And of course, in practice, it's the actual realized yield that we're interested in. And if we do a bit of rearranging of that equation, we arrive at the equation at the bottom, which expresses alpha, which is our measure of tolerance, the slope of the line, as defined by the difference in the health area absorption between a fully protected crop and what we actually manage to achieve in the field, and between the yield of a fully protected crop or the yield that we actually achieve in the field. And that enabled us to explore where the kind of optimum breeding target sits. So here we've got the solid line that represents the envelope within which it's feasible to breed, as represented by these particular populations of offspring of these wheat crosses. Now, if there was no disease, if we could completely eradicate disease from the crop, then the health area duration loss caused by disease would be zero, and we can use the equations we saw on the previous page to define a dotted line, which is a contour of achievable yield. And where that contour just touches the envelope is the optimum breeding target, in this case for the zero disease case. And obviously enough, if there was no disease at all, the thing to do is just to breed for potential yield and forget all about tolerance. However, the picture changes quite considerably even if we look at quite low or moderate amounts of disease that equate to a healthy duration loss of about 10 units. The contour of achievable yield then becomes the sloped dotted line that we see here, and where that touches the envelope becomes our new breeding target. And we can see that it's actually worth sacrificing a bit of potential yield in order to maximize what we can actually achieve in the field. So that's kind of defined the breeding target, but if we're farming, what can we do to exploit tolerance? And the difficulty is that since the work we did 20 years ago, there has been no work, or no consistent work across seasons and across modern varieties to quantify tolerance, and that's partly because it's quite costly and labour intensive to do so. However, if we take a look at the recommended list of winter wheat varieties here, this list does give us good data on the untreated yield of those varieties, and we've seen over recent years really exceptionally good progress in increasing the untreated yields, in other words, reducing the impact of disease on yield in the absence of fungicide treatment. And that untreated yield is to a large extent derived by the level of disease resistance and escape that has been bred into these varieties, and that has improved markedly. But these values are also influenced by the degree of tolerance to disease that's in these different varieties. So if we pick varieties with high untreated yields, we're actually exploiting to some extent the variation in tolerance between modern varieties. So what can we conclude? Well, wheat varieties differ significantly for tolerance. There is unfortunately no tolerance information directly for current UK varieties, but high untreated yields indicate varieties with good disease resistance and tolerance. There is a trade-off, as we've seen, between breeding for tolerance and breeding for potential or fully protected yield, and breeding for a combination of potential yield and tolerance is actually what's needed in order to maximize the actual realized yields that we can achieve in the field. And if you'd like to know more about this subject, there are quite some publications to look at.